Hi everyone, welcome to another chemistry video. We're looking at colorimetry and UV visible spectroscopy, which is 18.4. Okay, so just briefly, spectroscopy is all about investigating molecules through their absorption of light. And so we know that light is electromagnetic radiation. There's a picture here of quite a broad spectrum. I might zoom in on that a little bit. And so you can see that you have frequency and wavelength, and they're kind of opposites. But we end up with radio frequency, which is quite big waves, which is why they've got the in their wavelength, it's quite big. So it's sometimes actually up to like a 10-meter wave potentially. We have microwaves, which you guys would use in your kitchens. Infrared visible light is down here, which has again the wavelength of 10 to the negative 6, so quite small waves relatively. Ultraviolet is at the end, and then X-rays, and then gamma rays, which are really small. And so when we're talking about spectroscopy, there's a couple of ways, but looking particularly at UV visible, it'll be using this area of the spectrum. When we are actually thinking about light, we need to make sure that we understand what we're talking about. So I'm going to look at this apple picture first. So when we see a red apple, it's not that the apple is absorbing red light, it's actually that the apple is absorbing, say, the green and the blue light from white light, and then anything around the red spectrum does not get absorbed, and so that's the color that reflects most strongly. So when it's red, it means that it's absorbing other wavelengths, and red is reflected. And so that's just basically make sure that you are aware of how the light reflection works. So I put here that absorption of light is directly proportional to the concentration. So you know when you have cordial and you know that you have a stronger cup of cordial, it's more red or it's more green or whatever. So the more cordial that you have, the stronger it appears. Similarly in colors, in concentration with things that are dissolved. So we know that a leaf is green because of chlorophyll. It does not absorb green light, but it's rather it absorbs red and purple, and therefore green bounces back. Okay, so we're looking briefly at a process called colorimetry, which is basically a simple way of measuring the intensity of color in a sample. So basically, you'll be using a filter to select the colored light, and you want to make sure that, again, you're aware that if I'm testing something that looks green, I want to be putting in colors that are not green, so primarily probably reds and purples. So that's the complementary color to the observed color. If I've got a metal, so a cation or anions that aren't colored, they can actually be converted to something that is more colored, which we'll look at again in our chromatography things when we're looking at metal complexes. So really good things about colorimetry, which is in this section here. So it's simple, it's inexpensive, it's accurate, and it detects most cations and anions, which is awesome. So it's a really simple way of doing things. And we've got to remember our Roygabov table of the colors, so red, orange, yellow, green, blue, violet. And then if you're wanting to find the complementary colors, so if something looks red, if you're shining blue light at it, that's probably the best for the best absorption. If something looks orange, green, yellow, violet, and then similarly backwards. So if it's green, it looks red, if it's blue, it looks orange, etc. So the way I think about this is you have your Roygabov, and if you cut it right down the middle, if you just want to find the opposites, you have Roy, and you put that to the other side. So it's GBV and then Roy. It's probably the best way to think about it. I think I've actually written that wrong, so don't get confused. That should not be like that. It should actually be a G and then a B, pardon me. So it's green, blue, violet, red, orange, yellow. Okay, so colorimetry is a process. It's got 5 main parts, which you can see here, which I don't think has come out great, but that's okay. So you can see that you have a light source, which is basically just white light, potentially. You have the split, which is just putting up like a little filter. That means that you can isolate the wavelength of light, so perhaps a particular color of light that you would like. You have a very narrow beam. Then you have your solution, which is your samples dissolved in say water or another solvent that works for it. Then you can have your electronic detector, which measures the intensity of the light that passes through the sample. And then you have the recorder, which gives you basically a number showing you how much light has been absorbed. So that's the basic process, which is pretty much repeated throughout the next 4 or 5 different processes. So it's just your light, you're narrowing that light, it goes through your sample, which is say in a glass tube, into a detector, and then gives you a reading with a number. Okay, so the big question I think then is, how do you calculate the concentration of your unknown? So what you have to do is actually something called calibration, which I write down the bottom. Calibration, which means that you are preparing your sample with a series of other samples that you know. So it means that you can draw a graph basically and read off your unknown against all the things that you know. And so it ends up being like a linear graph, a straight line, and you can situate your unknown because you have one of the readings and you just read off the other one. So step 1 is prepare a set of standard solutions, things that you know, of known concentrations. Step 2 is measuring the amount of light absorbed by each standard. So you do a colorimetry process for each standard, getting your absorbance reading. You've got to, including in that, you do your unknown. So you know all these different concentrations that you put in, and then you do your unknown, and you still get a reading for your absorbance. Next, you want to plot your calibration curve, so absorbance versus, which is on the y-axis, versus concentration on the x. So this one's probably the most important part of the whole thing. Absorbance versus concentration. Okay, you gotta know how to do that. Then you want to look at the concentration of the sample from the calibration curve. So again, this is just supposed to be a introductory video. So I'm just going to make sure that you're aware you need to go to page 469 to 470 and looking off a linear graph. It's really not that hard. So you'll see on the next slide that I've got an example for you. So here's like a typical kind of question. So you might be given your sample, which is your unknown. So you don't know how much concentration that is, but you do know that it absorbs at 0.38 for that concentration. And then it gives you a series of known concentrations and the values. So when you have 4 micrograms per milliliter, then you have 0.16, and then 8 similarly comes to 0.31, etc. And so you end up drawing your absorbance on the y-axis and your concentration on the x-axis. And you get a straight line, so a linear graph. And because you know your absorbance is 0.38, which you would be measuring up here, pardon me. So 0.38, you follow it to where your line is, because that's your rule. You measure down, and then you end up with it being about 9.4, potentially, micrograms per milliliters of Fe2. Right? So you'd have a series of samples of iron 2+, because you know that you're looking for iron, and then you simply read off the graph. So often the question might give you a table, and you just basically plot it, or it might give you the graph already, and then you just have to read it off. So not too tricky. So the second thing that we're going to be looking at in this video is UV visible spectroscopy. So in one sense, it's very similar to colorimetry, but instead of just using the visible light spectrum, it's actually using UV as well. So the first little question for you is, what color is this sample? So we've got our absorbance and our wavelengths, and so we've got a little helpful graph at the bottom showing where violet and red light is. And so we know that we have a dip, which means that it doesn't absorb very much, around violet, blue, and then as it goes to green, we get an increase, and at yellow and red, we have our really high level of absorbance. So that means it's absorbing yellow and red light. So hopefully you're aware that that means that the sample is probably blue, because it has very little light that is absorbed there. It means it's bouncing back, reflecting off that substance into our eyes. So for example, this could be copper sulfate, pardon me, which in solution is blue. So UV Vis is basically another way of determining the concentration of your substance, and it's always in solution as well. And so we know that the light absorbed of the unknown concentration can be measured against the standard solutions within a calibration graph. So it's exactly the same idea, exactly the same. You just need to do your known standards in the calibration and then read it off from your graph. For UV Vis, it's the only thing that's different is, if you can see here, you've got your light source, you've got your slit, and then you have something called a monochromator, pardon me, which basically lets you select light of an exact wavelength. So you can be really, really specific, which then is absorbed into your solution. Then you get your detector and your recorder. So it's very much the same. The only thing that's different is they've added this extra step, which means that you have a bit more control and a bit more accuracy. So it can provide accurate results, but it is more expensive, unfortunately. So here is a UV visible absorption spectrum of chlorophyll, and we can see that it absorbs strongly around violet and also at red. And so you see in this middle gap, that's where it is green, because it is literally not absorbing any green at all. So that's always going to be reflected back. Okay, last couple of things. So when we're looking at our uses, we know that it is used really a lot in medicine. So if you're measuring urine, blood, all that kind of thing, so you can look at the hemoglobin content, the sugar levels in blood. You can look at how much dye is in plastic, thinking a lot about the presence of metal ions, or even phosphate levels in waterways. So this might be particularly relevant for us because we would have just finished looking at how we could analyze water. And so you can see that you determine the phosphate levels by using UV visible spectroscopy if we had more machines, which would be awesome. Okay, so finally the last thing to think about is metal complexes. So some metals actually might need to be converted into highly colored metal complexes to analyze in the UV visible spectroscopy. So this one example, you can see that you have your Fe3+. It's really sort of like a light yellow, but it's not very prominent. And so that's a transition metal. We know that if we have something that would be, say, potassium, and honestly it would be sulfur, carbon, and nitrogen, but I can't remember what that's called. That doesn't really matter. So you've got your SCN, which is clear, and then you put them together and you get a aqueous solution of FeSCN2+, and it's blood red. So that's really going to give a really strong reading on the UV visible because it is so coloured. And so it's just being a note saying that some metals, if you want to detect them, you can. You just need to convert using a chemical equation or a chemical reaction. And some of those transition metals are very, very colourful, which is really helpful. So that's been our video.